This talk is about large-scale entanglement and emergent biomolecular processes. C.Jayant Praharaj, Band Photonics Materials. So this talk, uh, we basically talk about entanglement, quantum entanglement, and uh, biomolecular processes. This slide is just a uh, preliminary slide which shows uh, a specific uh, energy scale or time scale in the universe, and we have just focused on indium gallium nitride alloys, which is a semiconductor system that many people work on. And you can have non-relativistic and relativistic entanglement generation in this done through structure design and device design. So we are interested in how large-scale entanglement in the universe has implications for biomolecular processes. We want to see in certain energy scales what kind of entanglement can be achieved by human design. And that can give us some clues about the kind of entanglement evolution, for example, that can happen in a realistic universe, and how it can get into the energy scales for biomolecular processes. For example, biomolecules have many energy scales which are not very different from the where energy range is showed here. So that's why we show this as a preliminary slide. The next slide, we move on to a slightly more exotic quantum interaction regime, and we have quantum field theory of quantum chromodynamics, or QCD, for strong interactions, which is seen in nuclei. For example, the kind of forces that holds nucleons together, that holds protons and neutrons, many protons and many neutrons together in an atomic nucleus. And the origin of that, the fundamental origin of that is a strong force, and that has, for example, up quark, down quark, strange quark, and then there are three other types of quarks, and then there is something called color. So SU3 flavored quarks have coupling to non-abelian gauge SU3 colored gluons, and these up quarks and down quarks, they interact by exchanging gluons, and that's part of the picture of how the strong interaction works, and that manifests itself in the nucleus. Now, why are we talking about it? The reason is we are talking about long time scales and the evolution structure of the universe, and we want to also consider if there are implications of these other sectors. So these are high energy sectors. For example, QCD, strong interaction, and typically involves much higher energy than, for example, biological molecules. But can it have indirect implications for entanglement over time and for biomolecular processes? So here we just mentioned briefly that SU3 flavored quarks have coupling to non-abelian SU3 colored gluons. And then there are three types of colors of the quarks as well. These are just some details of this sector. But we must be focused on what this sector can mean potentially because of the long time evolution of the universe for biomolecular processes in the present day regime. Here we have some further modifications. For example, we have gauge interactions with gravity. And why are we going to gravity? Again, the thing is, this is responsible for the large scale evolution of the universe, and how, for example, we know the universe is currently expanding with rate of expansion. There is also a rate at which the rate of expansion is changing, and that has to do with things like dark matter and dark energy and things like energy, things like that. But the thing is, the long-term evolution has somehow gone and given rise to biomolecular processes, and we are interested in seeing potential implications of these sectors of quantum field theory for these biomolecular processes as well. Next, we move on to the actual dynamics of biomolecular processes. And So you have amino acid chains and local processes. So these are some local processes, and we should just mention here that these are so the typical energy scale is EVR electron volts. We have, for example, these amino acid chains which give rise to proteins, and these are just showing the details because ultimately these form the core of biomolecular processes happening in living organisms. And we want to know if uh, uh, the evolution of quantum entanglement over time gave rise to the kind of structure that is necessary to sustain these living processes and these biomolecular processes. These, for example, show primary, secondary, tertiary, and quaternary protein structures. And so you can see that it's quite an intricate mechanism, and it is all these kinds of structures, primary, secondary, tertiary, and quaternary structures that are responsible for the kind of biomolecular processes we see. The next state talks about general quantum states in living matter. So here we have, for example, what are called tensorial states. So psi1, psi2, and which are basically uh, these factored states. And then we can define what is called a density matrix vis-a-vis these states. And you can look at the total state, and you can look at the projection of the state into a subspace. And because sometimes you want to, for example, integrate out the effect of the environment. And so there may be an environment setups, a bunch of states here which represent the environment, and you want to integrate those out, maybe thermally, maybe some other way, and then look at the kind of structure that you have remaining in there. So the thing is, fundamentally, these are quantum states. The thing is, how quantum these are in actual manifestation depends on the details of the system. For example, you can have things like, for example, you have things like coherent transport, which is a very quantum manifestation. Or do you have things like interference taking on, which is a very coherent manifestation? And the other question is potential quantum computing going on? And there's a lot of research and some answers about whether quantum computing is going on inside a living material or not. The research is still going on. This slide shows an information transcription between states. And so you can think of these as biological quantum channels, and which have error probabilities associated with them. And so there are probabilities associated with which specific, for example, these three bases of DNA segment, which encodes for an amino acid, can get transcripted into different bases based on probabilities of error and things like that. So this is basic schematics for information transcription using the DNA structure. This slide shows protein dynamics timescales compared with galactic and de Sitter timescales. So so we have time scales here which range from femtosecond up to seconds, and then we have galactic and de Sitter time scales which are of the order of 10 to the power 15 years or several billion years, or even 10 to the power 16. And obviously, evolution over 10 to the power 16 years has given rise to um, these complex biomolecular uh, mechanisms that take place in living processes, and these quantum processes, which are taking place all the time. Now, they're, as I said, depending on how much interference component you can see and how much coherence component you can see and how much entanglement component you can see, it can be more or less quantum, but these are fundamentally quantum processes with some uh, happening in some thermodynamic background. And so uh, it is interesting to see uh, what implications galactic evolution and de-Sitter timescale evolutions have over this over the, for example, protein dynamics, which are of the order, which are of much smaller time. So the thing is, uh, as the universe is evolving, what kind of, for example, femtosecond to second time scale dynamics were taking place in the universe already? In other words, what is the rate at which femtosecond to second time scale evolution was taking place, and what kind of role, for example, different quantum fields like QCD, QED, and the electroweak interactions were playing in this kind of evolution. Here, this slide basically shows another detailed slide which shows that the role of dynamics in protein function can be explored using nuclear magnetic resonance, and the various time scales can be explored, and then we can compare them with galactic and de-Sitter time scales. Primordial evolution of life is basically this slide is basically an invitation slide for the rest of the slides, which here. Structures specific to life. At some entanglement structures that are specific to life forms and biomolecular processes emerged at some stage during the galactic, during galactic evolution. This kind of entanglement structure specific to life emerged, and this just shows a DNA strand. Next slide is another detailed slide which shows proton tunneling and enzyme catalysis, which is a specific set of mechanisms that take place and runs many of the living processes and biomolecular processes. And again, the setting for this came obviously from some kind of entanglement structures that evolved over time and settled down into a specific subset of entanglement structure taking place against a thermodynamic background. There are several interesting questions. For example, what is the fundamental role of thermodynamics, and what regimes can potentially be explored? That the science can always be in for surprises, and so what potential regimes can be explored in biomolecular processes vis-a-vis entanglement and the thermodynamic background. The slide shows something called DNA computing. This is not quantum computing, but this is towards quantum. So you can think of this is like a combinatorial computing that can be achieved using DNA, where it can solve several problems that quantum computers cannot always solve that classical computers, sorry, the classical computers cannot solve in short enough time. The DNA computing can, and then this goes towards quantum, which is obviously the computing setting which can really solve enormously complex problems in short periods of time. Short complex problems in short periods of time. This shows just another detailed slide about DNA computing, how it works, and this is going towards quantum computing. Quantum computing is different from DNA computing, but see that both offer, for certain kinds of problems, both offer immense speed up in computing power over classical computers. This slide shows quantum biology aspects. As I said, we are always interested in quantum biology aspects because the large-scale entanglement structure has somehow evolved into the entanglement structure in a thermodynamic background seen in living matter. How much of the quantum component is manifested, and how much of it is more of how much of it is, for example, coherent, and how much of it is incoherent with the thermal component dominating. So the quantum side, we have, for example, photosynthesis has long-lived vibronic coherence, which could help solar energy. Enzyme catalysis is proton tunneling, which is quantum in nature. Olfaction, mutation, magnetonavigation are all regimes where some quantum phenomena are seen or can potentially be seen. And then the thing is, many scientists that think that the brain may have specific quantum features that are not necessarily captured by merely by just biomolecular thermodynamic processes, that there may be quantum aspects to it. And so these are all potentially related to how quantum fields have evolved over time. This slide shows a photo-induced catalytic activity. So this where it use quantum dots, artificial quantum dots, to photocatalyze certain biomolecular processes. And again, the thing is, partly artificial, but similar natural processes are happening. And what is the implication of large scale space-time evolution and quantum evolution on these biomolecular processes? Next, we move on to certain other interesting aspects of biomolecular processes, because again, we want to understand if there is anything unique looking about biomolecular processes, not completely unique, but very unique looking. How are they related to large scale time evolution, space time, and quantum fields? Superfractals, which are random fractals and linear structure. So you can see we basically have fractals and its projections onto a certain lower dimensional space, like a two-dimensional space. Here people have achieved fractal molecular assembly. The thing is, you can see that there is fractal structure, as there is self-similar structure. And so the field of mathematics of fractals is kind of developed, but there's a lot of open questions out there that people are still trying to prove and investigate. So here people have grown fractals. So there are different regimes of growth molecules on substrates. For example, 2D crystals, or you can have 1.6D fractals or other kinds of fractals of different dimensionalities. This slide basically shows how life forms tends to organize, and in the process of organization of life forms and the biomolecular processes that the static and the dynamic aspects, the structure and the dynamics aspects, you can see fractality. Interesting question is this fractality coming from some fractality which is fundamental in, for example, um, theories of potential theories of quantum fields. For example, some people have explored uh, theories of quantum field theories in fractal settings, and uh, so those fractal components of the field theories can have can have projections in this sphere of biological processes or living processes. Slide just shows a synthetic fractal molecule with iron and ruthenium, which forms fractal molecules. So fractality uh, is of interest when you talk about biomolecular settings and biomolecular processes and the fields and the setting in which biochemistry takes place. This slide moves on to something called Calabi-Yau biomanifolds, and basically there's something called string theory in physics, which basically tries to solve some mathematical problems associated with common field, quantum field theories, which have so-called infinities. And then they introduce strings and higher dimensional uh, settings in which these infinities can be read off, and good mathematical results can be obtained that can be compared with experiment. So in the biological domain also, the string brain theory has some potential applications, and people have explored these. This shows, for example, whenever the higher dimensions have to be compactified into a lower dimensional setting in many of these string brain theories. And in the process of doing so, there are complex Calabi-Yau manifolds that are involved. And they're there in the fundamental string theories of quantum field theories, but they are also there in the biological setting. This slide shows well, prion molecules and then some incipient Calabi-Yau manifolds. Basically, continuation of the previous slide, showing how Calabi-Yau manifolds can play a role in, for example, formulating a quantum theory for many of these biological macromolecules and biomolecular processes. And this slide shows from the fundamental physics, quantum field theory perspective, some common Calabi-Yau manifolds that are used for compactification. This slide shows, again, some more physics. These are some of the parameters involved in delineating the different kinds of Calabi-Yau manifolds that emerge while trying to get compactification in field theory. Basically reducing higher dimensional, more than 10-dimensional theories to effective 4-dimensional theories requires these Calabi-Yau manifolds. And these are all the different kinds, these are some of the different kinds of Calabi-Yau manifolds that can emerge, and you can see that there are numerous. So that's an interesting open question about how many Calabi-Yau manifolds are relevant, and whether there are other ways of doing this kind of physics and their potential applications in biology. This slide shows some further advanced compactification mechanisms for reducing higher dimensional theories to effective lower dimensional theories. And they have potential similarities with biological systems and biological quantum strings. These are called toroidal orbifold compactification, which use some more complex topological structures. This slide is basically kind of, it has been shown in the previous slide, this UCD as interaction regime. We are obviously mentioned that ultimately these interaction regimes lead to, for example, this kind of biomolecular processes, or this kind of biomolecular processes, proton tunneling, or photo-induced catalytic activation of nitrogenase. So these ultimately fields involving curiosity, although we are more interested in quantum, one would argue that quantum electrodynamics or QED is more relevant. But there are questions like the kinds of molecules that are seen and whether there could be relics from some of these other kinds of fields onto the biomolecular world as well due to quantum evolution over time. This slide talks about black holes and information because you can think of biological processes also as a kind of quantum computation and about processing of information. There are people who have looked at it, and these slides basically talk about this, goes in a little bit detail and not beyond the main focus of this talk. This is a Schwarzschild equation for a black hole. This is the Schwarzschild radius, 2gm over c square, and this is the Bekenstein-Hawking entropy. And then there are questions about if there is entangled. Uh, so the so uh, question during and after black hole formation, what happens to entangled state into forming black holes, for example, is a question that arises. The thing is, why would we be interested in that in the biological regime? And the answer is, it has been the galactic evolution and the evolution of the universe has taken place over a long time, and uh, the stars around which planets uh, are generated tend not are not always first generation stars. They are second, third generation stars. And so what happens to information over time, and how did, for example, a planet like Earth, which has living beings and biomolecular processes happening, what are the implications for this large scale evolution over uh, the biomolecular processes that are taking place, and the kind and amount of entanglement that is relevant, and how much entanglement went into, for example, generating potentially biomolecular processes and in running them over time, and what is the future of this. This slide moves on to even more physics, white holes and wormholes, and towards biological information. And this basically shows how a so-called Penrose diagram of a black hole with horizons and singularities, and how you can go from a black hole to a white hole which spews out all the material that is in it. And again, what happens to kind of setting and information in this kind of setting, and biological information over large time. This slide basically shows just another detailed slide showing something called cosmic microwave background, which is a relic of the Big Bang theory of the universe. And these are some details that physicists have calculated for fluctuations seen, multiple fluctuations seen uh, in the cosmic microwave background, how it agrees very well with theories. The experiment agrees very well with theory, and then the anisotropy of the cosmic microwave background that people are studying. This could also have potential implications for entanglement evolution and biomolecular processes. So in conclusion, the entanglement structure seen on a large scale in space-time leads to specific entanglement and pseudo-entanglement in living matter. And scrambled configurations can result due to measurement sequences and various length time scales. Fractal structures during evolution can emerge from mathematically specific kinds of entanglement, for example, those based on intrinsic fractal dynamics. And entanglement and thermalization of biological variety are key to evolution and possibly cyclic evolution. So that is the basic talk about entanglement and emergent biomolecular processes. Thank you.